Greetings everyone. This week, or this video, we're going to be talking about the cell structure of bacteria, archaea, and eukarya. Now, this might not be covered by your professor. In fact, last quarter, none of the professors went over this. However, I genuinely think it is really important to know this because otherwise things will not make sense. And I saw that with a lot of students last quarter where they didn't really understand why certain things were happening. And what I realized is it's because the professors did not go over the cell structure. So this may or may not be part of your curriculum, but I think it's really important to understand and have a good basis on of how the cell is laid out and the basic what you can say, like the functions of each of the parts of the cell. Okay, so let's break it down first into like kind of bacteria and archaea, what we call the prokaryotes. The prokaryotes. Okay, and then we'll talk about the eukaryotes pretty soon. Remember, this is not designed to be a very like in-depth, everything is covered sort of thing. It's just the big parts of everything, that I think is the most important thing to know, especially if you're not going over it. Well, then you don't need to know the in-depth things. You just need to know the basics, so that you can understand the rest of the cell. So these prokaryotes, bacteria and archaea, they lack membrane-bound organelles. Meaning that all the membrane-bound reactions that take place in the cell occur in the cytoplasmic membrane. Okay, this will be very important when we start talking about photosynthesis and cellular respiration. It does not take place in organelles or anything like that. It takes place on the actual cell membrane on the cytoplasmic side. Okay. The genetic material is located in a self-defined area called that should not say nuclei, it should say the nucleoid. I'm guessing it like auto-corrected when I wasn't paying attention, or I wasn't paying attention and Google auto-corrected it when I was typing this up. So, sorry, it should be the nucleoid. Okay, not the nucleus, not the nuclei, not the nucleolus, but the nucleoid. Okay, and that is right because prokaryotes do not have membrane-bound organelles. The nucleus in eukaryotes is a membrane-bound organelle. So by definition, they cannot have a nucleus. They have a nucleoid, which is essentially just a spot in the middle of the cell where all the genetic material is concentrated. Okay, and now this genetic material, what we call like the chromosomes, right, is often just a single covalently closed circular. Remember what covalent bonds are. It's circular, but it's also double-stranded DNA molecule. So oftentimes single, circular. That's basically the basics. There are some exceptions, such as bacteria can have some linear chromosomes. And then bacteria and archaea can have plasmids, which are kind of groups of genetic material that can code for things or not code at all. It's kind of a little bit of a gray area, but we may cover plasmids, we may not. I haven't decided yet if I want to, but they would be in another video. Now, some bacteria, some archaea have a cell wall. It's not like they for sure have it, they for sure don't. You'll find a lot in biology where there's not really definite rules. It's basically rules are made to be broken. It's more of general guidelines. So some have a cell wall, some don't. It's not a perfect way of distinguishing bacteria, archaea, and eukarya. Most of the time, though, they're relatively small compared to eukarya. So eukaryotes are typically the largest cells. There are a few exceptions, like always, where the bacteria or archaea are very large and larger than a eukaryotic cell. But in general, they're relatively small. And now what I want to draw attention to is, what does this mean in terms of like the transport of material, right? If you're very small, how do you transport things inside of the cell? And once it's inside of the cell, how do you transfer things from one side of the cell to the other, right? Because you're just getting into the cell, like a nutrient is one part of it. You need to be able to get it around the cell. So if you're very small, what sort of mechanisms would you use? And would you be able to use that same thing for eukaryotes? So to answer this, we're going to look at cell size versus the transport. So bacteria and archaea are relatively small. Now what this means is they can rely on a process called diffusion for substance transport. Now what is diffusion? Diffusion is essentially a spontaneous process where a substance will travel from a high concentration to a low concentration. Okay, so meaning, let's say we have a semi-permeable membrane. This will come into play later. We'll talk about it a little bit today, but mostly when we talk about cell membranes. But so a semi-permeable membrane, what will happen is we'll have a bunch of the substance over here, okay? And not that much on this side. What's going to happen is the molecules on the right side are going to keep bouncing into each other, right? Random molecular motion. We'll talk more about this in chemistry, okay? And so what's going to happen, they're going to keep hitting each other and hit each other through the membrane, right? And they can pass through. This is a semi-permeable membrane. We're saying this membrane will allow through this green substance. They'll hit each other. They'll move over, causing a gradient to form. And these substances move across the gradient. And gradually, this area of high concentration on the left will decrease in concentration. This area of low concentration on the right will gradually increase until they're about equal. Once they're equal, they will still travel through the membrane, but there will be no net difference. So there's a difference a difference in meaning between net and what's actually happening. So essentially, at this point, when the sides are equal to each other, we would call that equilibrium. So, of course, in our drawing, we don't have equilibrium. It's still traveling from high concentration to low concentration. This is just random molecular movement. Doesn't require energy. It just happens. Okay? And I was kind of talking to you guys how that through. The molecules are hitting each other. Since there's a lot less on this side, they're less likely to hit each other, so they're more likely to stay here. And like I said, this will go until the sides are equal, and then there will be no net change. Now, this will pose an issue for the cell that we will talk about pretty soon when we start talking about metabolic pathways. One thing, though, diffusion does depend on viscosity, or the thickness of a solvent. So as you increase the viscosity, you decrease the efficiency of diffusion. The way I can explain it is, how easily are you able to travel through water versus honey? Okay, traveling through a pool of honey would be terrible. It's super, super viscous. It would be really, really hard to travel through it versus water. I mean, that's our swimming pools. We do that all the time. The opposite of viscosity is fluidity, right? You increase the fluidity, the more it flows. So you don't need to know really the technical definition of viscosity. You just need to know that, it basically is like the thickness, right? It's, think of a very viscous thing as like honey. It doesn't really flow much. And when you do that, right, you it's very hard to travel through, so you decrease the efficiency of diffusion because the molecules are kind of stuck, rather than being able to easily flow through and hit each other. Okay, so now let's talk about eukarya. How we typically define eukarya is really the main thing, membrane-bound organelles. That's a huge thing. There's multiple other ways to define a eukarya, but in my experience, the best way is just membrane-bound organelles. So, what does this mean? Well, when you have membrane-bound organelles, rather than everything just happening, so if we were to draw a prokaryote, everything just happens here, right, in a prokaryote, in the cell, just anywhere in the cell. Of course, the things that require cell membrane will occur on the cell membrane. But otherwise, it just kind of happens in what we call the cytoplasm, or the cytosol. Really, there's technically a difference between the two. Cytoplasm is like the space. Cytosol is the substance that's in the cytoplasm, but in this class, we'll use them interchangeably. In eukarya, on the other hand, so if we draw a eukaryotic cell, things are compartmentalized, meaning you don't just have everything happening in the cytoplasm. You'll have some things occur in the mitochondria. Some things occur, maybe if you're a plant cell, you have it in the chloroplast. You have the Golgi apparatus. You have, let's see, I don't want to draw the Golgi apparatus. I don't feel like changing color. You have the Golgi apparatus. You have the nucleus. I'm not going to label them because we'll talk about them a little bit more in general. But the Golgi apparatus, you have the ER, which is like here. Okay. You have all these different things that compartmentalize these different processes into essentially a concentrated area. So you have all your ATP synthesis occur in one place, that sort of thing. Okay. But also think about it, right? If we were to show a very gross overestimation of a prokaryote versus a eukaryote, a prokaryote might be that big, and a eukaryote, might be that big. For the prokaryote, getting a substance across is pretty easy using diffusion. For eukaryote, that is a long way to travel, right? So there are now transport systems using microtubules, which we'll get into in a later video. So microtubules, microfilaments, all that fun stuff. Kinenes, dyneins, motor proteins, all of that we'll get into later. But it's because eukaryotes are so much larger. They need a much different and much more robust system of transport that prokaryotes don't need because they're so much smaller. Okay. So now what do all three of them share? Kind of the big ones, cytoplasm and cytosol. Okay. It's essentially the area between the membrane and the nucleus and the nucleoid, right? It's like a jelly-like substance. That's the cytosol, and it's like 70 to 80% water. That's not really important, except it's, if we remember what we talked about in the previous video, it's polar, right? Water is polar. So if this is mostly polar, then, well, if it's mostly water, then the cytoplasm is going to be polar, right? Okay. They also all share the plasma or the cell membrane. You'll use those. You'll hear those used interchangeably. Ooh, words. And it is semi-permeable, meaning that some things are allowed through. Some are not. Permeable means to pass through. Semi-permeable means, well, semi means not quite, so it's not quite able to pass through, meaning that some substances can easily go right through, some things cannot, some things require assistance. There's a whole That's why we use the term semi-permeable, and that'll come into play once again when we talk about metabolism and when we talk about cell membranes. And really, the cell membrane just separates the cell from the outside and controls what can come in and what goes out. So there's multiple functions in the cell membrane. We'll talk about it, what it's composed of, in a future video. So other things they share, they have the ribosomes. Okay, this is protein synthesis. Every organism needs proteins. In fact, one theory is that before life evolved, proteins had to come first. Okay, so the proteins came first, the amino acids, and proteins are made of amino acids, so the amino acids came first, then the proteins, and then from there, we got our genetic material, everything else, cells. That's just one sort of theory, and it logically makes sense. I haven't kept up on that because I have my own things to keep track of, but much smarter people than me are studying that and really figuring out how life came to be and how life evolved. Well, we know how life evolved, right? Evolution, but how life came to be. And there's like 4 locations where you can find these. You can see them freely floating in the cytoplasm. These are just called free ribosomes. You can see them attached to the cytoplasmic side of the cell membrane, and these can kind of make proteins for the cell membrane. You can see them attached to the outer membrane of the nuclear envelope. Once again, it makes proteins. The location kind of depends on where the end result is or what the purpose of the protein is. Or you can find the ribosomes. I just realized I had freely floating on here twice. I should have proofread this before. Anyways, it can be attached to the cytoplasmic side of the endoplasmic reticulum, which is something we'll talk about in a little bit. So the 2 main ones you'll see are freely floating and attached to the ER. The other 2, not so much, but it's worthy of noting. The one place where you won't find them, notice this, is you won't find them inside the nucleus. So even you could say, well, prokaryotes don't have it, but in eukaryotes, you won't see them inside the nucleus. In fact, I remember on my first or second, I can't remember which midterm it was, there was a question, and one of the wrong answers was that there's a ribosome in the nucleus. They're in the nucleolus. I don't remember. But anyways, there are no ribosomes there. They're mostly in the cytoplasm. Well, they're all in the cytoplasm, just kind of where in the cytoplasm depends. Okay? So now we're going to take a little bit more in-depth look at Eukarya. Not all the way, because there's still some more things we're going to get into. But so the nucleolus is the area within the nucleus that assembles the ribosomal subunits. No, it's not where the ribosomes are, it's where they're assembled. Okay, so they assemble the ribosomal subunits, then they leave the nucleolus, leave the nucleus through nuclear pores, and then when they attach to an mRNA, which we will talk about in translation, it becomes the completed ribosome, and translation takes place. Now, this is just done in the cytoplasm in prokaryotes, but remember, what is this called, right? It's in the nucleus versus the cytoplasm. This is the compartmentalization. Well, another example of compartmentalization. Okay, next we have the mitochondria. This is only there in eukaryotes because of how the mitochondria came about. Okay, so this is the primary site of metabolic respiration, and note that it actually has its own DNA. So not all eukaryotic DNA is in the nucleus. Some of the eukaryotic DNA is outside of the nucleus in the mitochondria. And if you're a plant, which we'll talk about in a little bit, you will have some DNA also in the chloroplast. But that's a tangent. We can be aerobic in the mitochondria. And aerobic just means oxygen is the final electron acceptor. But it can also be anaerobic. Oxygen is not the final electron acceptor. In humans, we strictly have an aerobic mitochondria, okay? If we do not have oxygen, it's not like we can use something else for the final electron acceptor for in the mitochondria. We kind of just die. But there is another method we can use. It's called anaerobic respiration. It bypasses the mitochondria and does something else. But we will talk about that when we start talking about metabolism. Okay. One more thing, which was another question on one of my midterms, is the number of mitochondria in a cell can actually change depending on the conditions. Meaning that if you are a runner, okay, you need a lot of energy. You can actually have more mitochondria in the cell than someone who is not a runner, right? Who doesn't exercise. So if you are, and even that gets down to the individual cells. So like muscle cells might have more mitochondria than, I don't know, what's a cell that doesn't really do much? I can't really think of anything. But it's noted that like muscle cells and athletes tend to have more mitochondria than the rest of their body, or compared to like a standard cell. So you can get more mitochondria if you essentially like train your cells that, hey, they need to produce more energy. You use more, or you have more mitochondria, and you use more mitochondria. Next, we have the lysosomes. These are really just enzymes that aid in the breakdown of macromolecules. There's a lot more to lysosomes. If your professor wants to go more in depth about them, they can. But really, you don't need to know if your professor is not going to go over them, you just need to know that they can aid in the breakdown of macromolecules. That's about it. Now, vesicles and vacuoles, these are going to be membrane-bound sacs that function in both storage and transport. So vacuoles are more of the storage side. Vesicles are more of the transport side. And so these vesicles can actually like fuse with other components of the cell, meaning that if you have a cell membrane here and you have a vesicle coming, it can actually fuse with the cell membrane. And what will happen is you'll get something that looks like this, where this part here was the vesicle. Okay, so it can fuse. Now, vacuoles actually can't. They're much more for storage. And so in plant cells, we have what's called the central vacuole, which does a lot of things that you may learn about if your professor goes over it. But really just the main takeaway I have is it helps regulate the cell water concentration and then also provides some structural support to the cell wall. Yeah, once again, this is kind of a thing where if your professor decides to go over it, they may not. It's kind of up to them. As much in detail as they go or your textbook goes, that's how much detail I would know. Next, we have the centrosome, which we will talk about way in the future when we do, what's it called? I'm totally blanking on what it's called. Oh, DNA replication. The centrosome plays an important part in DNA replication. But it's where all the microtubules in animal and yeast cells originate. That's kind of like the preliminary thing. Like I said, centrosomes are really important for DNA replication, but we will talk about when we get to DNA replication, which will be for your class probably around week 6-ish. I can't remember. Maybe week 5, week 6. Okay, so plants also have a cell wall, right? Animals do not. Plants do. This protects the cell, provides structural support, and gives shape to the overall plant cell. Fungal and protist cells also have this. Notably, though, the bacterial cell walls. So if the bacteria has a cell wall, it's mainly composed of a component called peptidoglycan. If you've taken Biz2C, you would know what that is. Plant cells, however, are mainly composed of cellulose, which is a polymer of sugar. It's actually a very, very strong polymer, so humans actually cannot digest cellulose. Cows can in parentheses, because they have bacteria that secrete enzymes that break down the cellulose. And this bacteria exists in the gut of these cows. That's how they can eat like grass and whatnot, because they have these bacteria that can digest cellulose. But humans, we can't, and we do not have bacteria in our gut that can. They also have chloroplasts, so plants, chloroplasts. Animals don't have chloroplasts. That's kind of unfortunate, but oh well. And this is the site of photosynthesis, and like I briefly stated earlier, it does actually have its own DNA. The chloroplast is where the light harvesting takes place, and that occurs in the thylakoid membranes, and whereas sugar synthesis takes place in the stroma. And we will go a lot more in depth when we talk about photosynthesis and cellular respiration. So we'll talk about the ETCs, the mitochondria, glycolysis, and then for photosynthesis, the ETC and the Calvin cycle. Okay, so we're going to go a lot more in detail. We're going to draw out mitochondria and chloroplast so you can really see where each process takes place. And so now this kind of occurs basically because of chlorophyll. Chlorophyll is the substance that can actually capture light energy, and then the plants can use that to convert it to things like NADPH or ATP. So really quickly, the chloroplasts are hypothesized, and it's pretty good evidence that it was at one point a cyanobacteria that was engulfed by a non-photosynthetic cell. This is called the endosymbiotic theory. We will talk about, I think I'll do an entire video dedicated to the endosymbiotic theory. Finally, we just have a few more ones I want to go over. The endoplasmic reticulum. This is where you have the modification of proteins, and also you can get some synthesis of lipids. There's two types. You have the rough ER. This is an endoplasmic reticulum. That's what ER stands for. With ribosomes attached to a cytoplasmic surface. So the rough ER looks like this. It's attached to the nucleus, and then kind of comes out, and then like this. So if you've ever seen that sort of structure on a nucleus, that is the endoplasmic reticulum. What separates the rough ER from the ER is the presence of ribosomes. So they will just kind of dot around. There we go. So we'd call this one the rough ER. And then we call this one over here just the smooth ER. Okay. So the rough ER, right? It's an ER with ribosomes just attached directly to the cytoplasmic surface. This is where protein modification occurs. So this can be for destination in cell membranes or for secretion, meaning that proteins can both stay inside the cell or they can actually leave the cell. So secretion means for a cell to essentially get it out of it. So get a substance out of it, that's secretion. If a cell is secreting a protein, it's sending a protein outside of the cell. So if it's not going, but however, you can also have these proteins go straight into the ER, right? Because in order for all of this, you need proteins. So some of the proteins made by the rough ER will stay in the rough ER to help facilitate the packaging and modification for other proteins. Now, if it's not going to stay in the ER, it's going to be packaged into those vesicles we were talking about. So essentially what's going to happen in here, you have your protein drawn in blue, and then it's going to be packaged in a vesicle. And now this vesicle can be taken elsewhere, and we'll see what happens in a little bit. The rough ER also will help make phospholipids and will also be packaged in these vesicles, and those can be sent to the cell membranes or the plasma membranes. And the rough ER is made up or has a membrane, right? All the organelles in eukaryotes have membranes, right? That's what defines eukaryotes, right? Membrane-bound organelles. So that means they need membranes, so we need a place to make membranes. Now, I want you guys to think about liver cells have a lot of rough ER, which I believe this was another question on one of my midterms. So why would liver cells have a lot of rough ER? Well, think about the function of liver cells. Liver cells help break down substances in your body. Well, in order to break down, you have to have, what is that done by? Well, that's done by enzymes. Enzymes break down substances. So the liver cells has a bunch of rough ER because it needs to make a bunch of enzymes that are going to be secreted out of the cell. Remember, that's a very important part of the rough ER is making the proteins. Proteins are enzymes. Enzymes are proteins, not all proteins are enzymes. But so the rough ER helps make the enzymes, the proteins that you're going to secrete out of the cell that will then go and break down the substances that the cell can then absorb. That's why liver cells have a lot of rough ER. Okay, so now let's talk about the smooth ER. It has few or no ribosomes attached to the surface, right? That's smooth versus rough. It's kind of simple. It's based on what it looks like. And the job of the smooth ER is to, synthesize carbohydrates, lipids. There's also a few like sterile hormones, stuff like that. That is synthesized by the smooth ER. Okay, next we're going to talk about the Golgi apparatus. So when the rough ER makes its protein and it packages it in a vesicle, it's not done yet. It gets sent to the Golgi apparatus for final modification. This is where the vesicles are sorted by destination. They're going to be figured out, and this is all done through things in the RNA. Okay. So the genetic material will kind of tell the Golgi the Golgi apparatus where this protein is destined for. So it'll be sorted, it'll be packaged, and it'll also be tagged. So this is done through the addition or the removal of like sugars, or the addition or removal of phosphate groups. Okay, and we'll talk about phosphate groups in a little bit when we start talking about proteins and protein modification. We'll do an entire kind of thing on that. But so essentially, these tags will tell the cell where this vesicle containing a protein is destined for. So it could be inside the cell, could be on the cell membrane, in the cell membrane, outside of the cell. Okay, that's all done through this tagging process. And then I wasn't even sure if I should include the peroxisome. It was in my notes, so I'm just going to say it. But I literally have not had a single question on this. But basically, it just carries out the redox reactions. It'll produce hydrogen peroxide, peroxisome, peroxide, that sort of thing. Hydrogen peroxide is H2O2. Not that you need to know that. But so it carries out the redox reactions. It'll break down fatty and amino acids. And it'll also detox substances. So when you ingest alcohol, because, college students, I know you guys like to party. When you drink alcohol, the peroxisome will help detox substances by breaking it down. Okay. So, to end this off, I want you guys to draw a eukaryotic cell. Try to do it from memory. Think about all the things you need to draw. We should have gone over everything you need. And we're going to go ahead and say this is we'll say an animal cell. Now, the reason why I didn't just say draw an animal cell is because I wanted you guys, when you first saw eukaryotic cell, to be like, wait, I can't just draw a eukaryotic cell because there's multiple types of eukaryotic cells. That was the hope. But now I'm going to specify that I want you to draw an animal cell. So go ahead and do that. And I got to quickly do one thing. Can I actually pause? Oh, I can't. I will be right back. It'll be a second for you guys. Okay, I got done with my thing. Let's now draw a eukaryotic cell. So, what are we going to start with? Well, we need a cell membrane, right? No matter if you were drawing a prokaryote or a eukaryote, you need a cell membrane. Next, let's draw, let's say a nucleus. We'll put that right about here. We'll say this is the nucleus. Ooh, I'm actually gonna here's the nucleus. Okay, inside the nucleus, we have, right, a concentration of genetic material, which we're going to call the nucleolus. We need the nucleolus, not necessarily the concentration of genetic material, but where the ribosomes are manufactured. Okay, so we have the nucleolus. Then let's also, very quickly, let's just draw in some genetic material. Kind of, doesn't really matter. We'll talk more about DNA in the future. And we'll call this chromatin. Okay? And we'll get into chromatin, chromatid, chromosomes, all that stuff when we talk about DNA. Okay? So now, off of the nucleus, remember, this membrane here is semi-permeable. It's the nuclear envelope. Okay? Off of this, let's draw, I need a new color. How about this guy? We're going to draw the ER, okay? Both the smooth ER and the rough ER. How do I want to draw this? Okay. Da-da-da-da-da. Okay. Now we can draw a lot more. I'm not going to, just because for time's sake. Just kind of gonna go like that. That'll be good enough. Okay. So we're gonna label this one as the. Let me get a smaller. So this here's the smooth ER. And this one will be the rough ER. But what do we need to do? Well, we need to make it rough, so we'll add. Actually, that is a little too big. We'll add some ribosomes onto the surface. And this will be, right, what makes it a rough ER is the presence of ribosomes on the surface of the endoplasmic reticulum. There we go. So now we have our smooth ER and our rough ER. What do we need to do next? Well, we know that's not the only spot where we can find ribosomes. So we're going to add in some ribosomes around the cell. Just kind of go wherever. We're just going to call these the free ribosomes, right? It's not really attached to anything. Draw an arrow, and we'll say, actually, we don't even need to do that. Just say the free ribosomes. Okay. What else? Well, What happens when something's done in the rough ER? Well, we got to send it to the Golgi apparatus. Whoops. So let's draw that in red. Okay. So the Golgi apparatus looks like this. I'll put it up here. Kind of, whoops. Da-na-na-na-na. Okay. This here is the Golgi apparatus. So label it as the Golgi apparatus. Okay. Next, what do we need? Let's start. Well, we said it's an animal cell, so do animal cells have chloroplasts? No, but what do they do have? We know they have a mitochondria. So the mitochondria, and also I did not expect you guys to, get these perfectly right, these looks. I just wanted you guys to be able to, well, maybe I draw this, and then this is the ER, stuff like that. And now I'm showing you kind of what the actual structures look like. But so let's draw a mitochondria. And we only need to draw one because we're not saying this is an absolutely what it looks like. We're just, getting a basic general structure of the cell. So the mitochondria has these inner folds, okay, which we will talk about why they're important when we start talking about metabolism. So there's the mitochondria. Let's go ahead and label it. Mitochondria. Perfect. And now let's draw maybe let's draw a vesicle. Okay. So we can kind of draw a vesicle coming out here. Maybe it's getting transported somewhere. We don't know. We're just going to draw it just so we understand that it's there. But we also need to draw a vacuole. Okay. Vacuole shapes are kind of do a lot of things. We don't need a central vacuole here, right? Because we're not a plant cell. But we still will have a vacuole. Okay. And then finally, I'm not going to bother drawing the peroxisomes because, like I said, I don't really think you're going to have anything on it. But one thing we should draw, let's draw a lysosome. So I need a new color now. Trying to remember which colors I used is terrible. Don't think I've used this one. So we'll draw this. Just draw a few lysosomes. Okay, and this here, I'm going to redraw or rewrite cell membrane. That way it matches so my OCD doesn't freak out. This here is the cell membrane, right? So this is our basic structure of a eukaryotic animal cell. To make it a plant cell, we just need to draw maybe a chloroplast and a central vacuole and a cell wall. But we're just drawing an animal cell. Don't need to do anything more. And then also you should be able to draw a prokaryotic cell. So bacteria, archaea, that sort of thing. And understand what do you not have in a prokaryotic cell that you do have in a eukaryotic cell, and what do you have in a prokaryotic cell that you don't have in a eukaryotic cell. So kind of, the differences between the two. That was the idea of having you guys draw them out. I think if you can draw these processes and structures and everything out, I think you're gonna, and be able to label them too, I think you're gonna do well in this class of being able to conceptualize where everything is happening and how it all fits together. My biggest thing in this class is making sure you guys understand how everything ties in, okay? Because I've seen a lot of students fall into the trap of memorizing every little thing as its own thing, but not understanding how everything fits together. In fact, kind of my final exam practice that I sent to the students last quarter, it was 3, in my opinion, really, really difficult questions. And they all had to do with combining everything together. So that's gene regulation, mutations in your DNA, errors in your DNA, transcription, translation, what else? Protein function based on its structure. There was like 6 or 7 different topics. I think even one of them involved like cellular replication. So being able to understand how all these different topics work together to get you this end result of a functioning cell is going to be really important in this class. Maybe not in terms of the actual test questions. You will get ones that bring in multiple topics, but nothing like the questions all throw at you guys. But rather, I think it's just easier to understand the material if you understand how it all works together. But past that, that is all I have for this video. Like always, good luck with your studying. I know you're going to do great. Good luck with all the rest of your classes, and have a good rest of your day.